Hello, my name is Aurélie Lacroix. I am a research associate and Marie Skłodowska-Curie Fellow working at Sixto Bioscience in London. When the Berlin Wall fell, I was actually 5 months from being born. I actually wanted to be a singer, but I very quickly realized that this was not how I would make the world a better place. It would probably have make it worse. But I'm very happy to be a scientist, and I hope that my effort combined with the scientific community effort will benefit society one day. Chemotherapy causes so many side effects that some patients even refuse to take their cancer treatment. So today I'm going to explain you how we are building the next generation of therapeutics so that patients never refuse to take their treatment anymore. Against cancer, many drugs already exist, and they are very effective at killing the cancer cells. But they do have a problem. They also kill all the healthy cells on their way to the tumor. This is what causes so many dramatic side effects. People lose their hair and they get extremely sick. So, in order to address this issue, we, as well as other researchers, are developing what we call therapeutic nanoparticles. Those are tiny little objects that are able to encapsulate a drug molecule in it and bring it specifically to the cancer cells. So that healthy cells are not affected anymore, side effects are reduced, and treatments are more efficient. But obviously, I would not be talking to you today if it was as simple as that. It's been now decades that these revolutionary treatments against cancer are being developed, but they often fail when they reach clinical trial. What happens is that when a nanoparticle is injected into the body, it does not just circulate, it will first encounter everything that you have in your blood, red blood cells, but also thousands and thousands of different proteins. Those proteins come and coat and stick the nanoparticle surface and form what we call a protein layer. So now your body does not just see a nice nanoparticle, it sees a big mass of protein. And the targeting ability, the ability to reach only the tumor cells, is reduced. So when we looked at that, we thought, all right, we have basically two possibilities. The first one is to try to find a way to remove and prevent the formation of this layer, or maybe we could see if we could turn that problem into our advantage. And this is what we did. We chose solution number two. Indeed, what we knew is that there are proteins in the body that naturally help the accumulation to the cancer cells and that naturally help in reducing side effects. So we can introduce chemical modification that make our nanoparticle attract only the protein that we want. Basically just like a magnet. So we first had to find ways to study efficiently and quickly the formation of this protein layer and then do a bit of chemistry. With that, we can now make a nanoparticle that attracts only the protein that will help the accumulation to the cancer cells in order to reach our goal, which is delivering the drug exactly to the right place. So we believe that with this, we can help a therapeutic nanoparticle finally being brought into the market, as well as help both patient and doctor fight against the disease more efficiently. And we hope that one day, cancer treatment can be simple, safe, and allow patients to live a normal life. In this research, we are not making just another new nanoparticle. Instead, we are looking at one of the downstream issues that has been delaying the field, and we're trying to address it. So by understanding better how blood components interact with those nanoparticles, we hope we can provide tools to us, but also to other researchers in order to design our nanoparticle better and really make them a clinical reality. It is estimated that 1 out of 2 people will develop cancer through their lifetime. So knowing this, we know that we need tools for prevention, for diagnostics, but also to cure cancer more efficiently. Currently, classical treatment against cancer very often involves cycle of chemotherapies, radiotherapies, and maybe even surgeries. So our goal and hope is that we can make cancer treatment simple, safe, and really allow faster recovery for each of the patients. So we need to introduce chemical modification within our nanoparticle to attract the protein that we want. With the current system that we're using at Sixfold, it's very easy for us to put any sort of modification. But we have to choose among a huge library of different molecules. So we have to go through a quite time-consuming iterative process where we synthesize and then do different tests. On the other hand, because the area of research is quite new, there is a lot to discover. So there is also potential to impact on other fields than just cancer research itself.